Hello guys, this is Anuradha. So today I'll be teaching you Unit 9 Biotechnology of Class 12 NCERT. I would like you guys to open the biology textbook and keep beside you, page number 193, Unit 9. So let's begin. So, what is biotechnology? Some of you might know what it is, and some of you might not. So, I'll give you a brief introduction. So, to start with, does anyone know how do we get curd from milk and how do we prepare wine? All these are actually microbe mediated processes, I can tell. So, when I say microbe mediated, there is some play of microbe in it. That is any kind of microorganism to say, like bacteria, fungi, anything. So, how do we actually make curd from milk? You know, how do we make curd from milk? So, this is a simple process. You would have seen your mom or dad, to keeping a bowl of milk and putting a small amount of curd to it, and then when you keep it overnight and continue in the morning, you will be able to see that the whole vessel of milk had turned into curd. So, how this is happening? So this is also because of a microbe which is actually present in the milk, called as lactic acid bacteria. You would have heard about this name, lactic acid bacteria, also called as LAB, lactobacillus. Lactobacillus. So this bacteria actually, releases some acid. Actually, it releases acids and partially digests the milk protein. It digests the milk protein. So by doing so, what it happens, it coagulates the milk. It actually coagulates the milk, and the milk turns into curd eventually. So this happens overnight. When you keep it overnight, this happens. This process takes place, and also usually this lactic acid bacteria is present in millions of amount in curd, and that's why we add a small amount of curd in the milk and keep it overnight, because this curd actually contains millions of lactic acid bacteria, and this process follows as I told. This lactobacillus will release some amount of acid, and this acid will partially digest the milk protein, and it will coagulate it, and it will turn into curd eventually. So all these is actually, I can tell, this is microbe mediated process. Similarly, wine is also prepared by fermentation of malted, some malted drinks, and then fruit juices as well. By fermentation of that, we get ethanol as the end product during fermentation. You would probably know that the process of fermentation. So this happens, and then we are getting wine. Similarly, also bread making also, you can tell. Bread actually, it is used by, it is made by using microorganisms called Saccharomyces cerevisiae. This is a form of yeast, Saccharomyces cerevisiae. So actually, what happens is that over here also the process of fermentation only takes place. The what happens? Fermentation takes place, and then the yeast actually helps in making bread. Like the end product will be ethanol and carbon dioxide, obviously. So bread is also made by using the microbe yeast. You can tell. And so all this is a microbe mediated process. So why am I telling this to you? So this is all a biotechnological process. If I tell, that is, you are using a microbe or the product which is obtained from microbe, that is acid in this case, and you are able to prepare a product, that is curd, by using this enzyme, or enzyme or acid that is produced from this microbe or the living organism. And with this, you are making a product which is useful for human being. So this is basically biotechnology. In simple term, it is, it is a technique like this which is used, which uses live organisms or the enzyme that you obtain from live organism. In this case, for making curd, we are getting acids, right? This is only making us curd. Eventually these acids only digest the milk protein, and it makes curd. So this is an example which I'm giving right now. So it's a technique. Biotechnology is actually a technique where we are using organisms or enzymes from organism, and we are making products which are useful for human beings. This is basically all about biotechnology. You can tell that all the microbe mediated process are biotechnological processes, but it is actually restricted to a certain environment. this micro-mediated process. There are much more to biotechnology as well. So next, I would like to introduce two other techniques like in biotechnology itself. So we have genetic engineering first, and then we have bioprocess engineering. I'll explain to you what it is. So biotechnology as a subject, it's categorized like, modern biotechnology. How is it actually, divided? Genetic engineering and genetic engineering and bioprocess engineering. Bioprocess engineering. So, what is this genetic engineering? What is this bioprocess engineering? So, genetic engineering is nothing like you have you know that all the living cells, like if imagine this to be a cell, this to be its nucleus, and you have chromatin material inside. This is the chromatin material inside. So, this chromatin material, as you know, contains, chromosomes, right? Chromosomes like these. These chromosomes will in turn contain what? These chromosomes will in turn contain, DNA or the genes, right? The genes which will have the DNA actually. So, you know that DNA, it is the blueprint of life. So this is everything that is, controlling the, whatever processes that is happening inside our body is controlled by this, the genetic material DNA, obviously. So when I am altering the, the structure of the DNA or the chemical nature of the genetic material, we call this DNA the genetic material. So whenever I'm altering the chemical nature of the genetic material of the genetic material. And because of this alteration of this genetic material, like you know that DNA actually contains four nitrogenous bases: adenine, thymine, guanine, and cytosine. So any of these, if I'm altering, like if I'm altering the base pairs which are present in this DNA, then eventually what will happen? The whole genetic material, like the code, will get altered, right? So we know the central dogma of biology. You would have learned this in molecular basis of inheritance. So the central dogma, CD, I'm writing it over here, states that DNA transcribes into mRNA, and mRNA will translate into protein, right? So by doing so, all the proteins which are being made is controlled by this code which is present in the DNA. So if I'm going to alter the chemical nature of the DNA, you know what will happen? The desired protein will not is not going to be made eventually. It will be some other protein which will be made by altering the chemical nature. So this protein or the enzyme, what you're getting as an end product because of this DNA, which transcribes into mRNA, and then mRNA translating into protein, this protein will, for example, say will control the phenotypic character or the external appearance of the organism. So if I'm altering that particular code in the DNA which controls the phenotypic character, then the phenotypic character of the organism will be fully changed, right? So this is what is genetic engineering. What I'll do is I'm going to chemically change the nature of the DNA so that the protein made is going to be different, and because of this protein will control the phenotypic character, and a very different phenotypic character will be exhibited. So like this we can genetically modify an organism. So this is done using genetic engineering, right? As we say, genetic engineering. So now I guess the genetic engineering part is clear for you. So now let's come to bioprocess engineering. What is bioprocess engineering? So for bioprocess engineering, usually you know that now I have explained you an example about making curd from milk. So when I'm making curd from milk, it was a micro-mediated process, obviously lactic acid bacteria, LAB, and with its acid only it coagulated the milk, and then it gave us curd. So like this, many other products are actually made using this technique, like antibiotics, and we also have vaccines which are being made. All these are micro-mediated processes, but in order to make this certain antibiotic or to make a certain vaccine, what will I need? I'll need a certain microorganism. So in that way, I should actually ensure that I'm using the microorganism properly, and I'm going to treat its enzyme or the product given by that particular microorganism, be it bacteria or anything. In case of antibiotics, obviously it's going to be bacteria. So the enzyme from the bacteria should be properly used, and it actually has to be properly treated, and then it will help us to get the necessary, the required antibiotic, what we are actually aiming for. So for that, I need a sterile environment where I can grow fully only the required bacteria, only the required bacteria. right? I need to grow only the required bacteria in order to obtain a certain antibiotic. Certain antibiotic, right? So to obtain a certain antibiotic, I need a required bacteria. So what should I do? I should maintain wherever I'm, growing these bacteria in large amounts in vessels or, fermenters. I have to ensure that it is only, only the desired bacteria, the desired bacteria. For that, what should I actually do? Eventually, what should I actually do, for making the desired bacteria only, for growing the desired bacteria only? I should maintain a sterile environment. So, whenever a chemical engineering process is taking place, I should ensure that I'm using a sterile environment so that there is no other bacteria or no other microorganism which is involved, and only the desired bacteria or the desired microorganism is going to be involved, so that I will be able to get the desired antibiotic. In this case, bacteria, certain antibiotic which I'll be getting. So, vaccine also the same follows. So this is generally bioprocess engineering. That is, you maintain a sterile environment in chemical engineering processes in order to get the desired product. So these two are the techniques which are actually involved in biotechnology. If I have guys, that's it for today. I'll meet you in the next video of part two biotechnology. Thank you.